Hello everyone, my name is Dylan Hamilton from C.A.R. Entertainment and welcome back to another AS1 biology revision video. Our last one on the topic of cells. We're going to be doing meiosis, and I'm not going to waste any time. Let's get straight into this. So, meiosis, now this is different from mitosis, don't get them mixed up. Meiosis is a wee bit difficult, but basically it's just essentially mitosis occurring twice. So, it's the mitotic cell division maintaining the state of ploidity or ploidy. As I have it spelt. Basically, that's not really exactly the definition you want to know. You basically want to know that meiosis, or meiosis is the process of reduction division, where cell division halves the number of chromosomes. So, its process is that it takes place in the reproductive organs with 2 divisions and 4 daughter cells, with the chromosomes being arranged differently from each other and different from the parent cell. Now, interphase and prophase 1. Now they're going to have 1 after them. There's going to be prophase 1, anaphase 1, and then they're going to have 2. So we're going through twice. That's why I'm saying it's like mitosis twice. So interphase, you'll see your chromosomes there, and you'll have 2 centrioles. In prophase 1, there'll be a homologous chromosome forming bivalent. Now 2 chromatids and will be there with your centromere, like in mitosis, and your nucleolus will be less obvious. Now, your bivalent will occur whenever crossing over happens, and this is where 2 chromosomes of this homologous pair will align, resulting in the bivalent consisting of 4 chromatids. Okay, 4 chromatids. So basically that's saying chromatid, centromere, chromatid, so there's 1 made together, and then you'll have another one in the same part. So then you'll have the point where these chromatids cross over is called the chiasmata. We'll now move on to metaphase and anaphase 1. Metaphase 1 is when the nuclear membrane will break down and the chromosome pairs will be aligned along the equator, just like in mitosis. The independent assortment may then occur, and this is where 1 chromosome from each homologous pair will enter the gamete. Anaphase 1 will then follow this, and this is where the microtubules of the spindle will contract, pulling the chromosomes to the opposite poles of the cell. Now with 1 chromosome, each homologous pair will end up in a daughter cell. So telophase 1 chromosomes will be pulled to the opposite end, each chromosome consisting of 2 chromatids. That's a key point. Cytokinesis is when 2 daughter cells each have 2 chromatids. Okay, so now we've went through it once. You'll have 2 daughter cells with 2 chromatids. Okay, so you haven't got we haven't got to the point where we're at chromosomes yet, which is the difference in between cytokinesis and meiosis and in mitosis. Okay, so you've got the same amount of daughter cells at this point, 2 daughter cells, but you've got chromatids still and not chromosomes. But then we go through it again. So you'll go prophase 2, centrioles will move towards the opposite poles. Then metaphase 2, where the spindles will form right angles to the plane and the chromosomes will move to the equator, attaching to these spindle fibers. Anaphase 2, the centromeres will divide and the sister chromatids will move towards the opposite poles. And then telophase happens again, where the chromatids are now become chromosomes, and the nuclear membrane will form around the chromosomes. So cytokinesis happens, and you have 4 haploid daughter cells produced, each with 2 chromosomes. So the significance of this: it's an essential part of sexual reproduction. It gives genetic variation among offspring, different characteristics. It's important for the survival of species. That's why people have blue eyes, green eyes, whatever, discontinuous variation, I believe it's called. So basically what causes the variation? It's crossing over, your reduction and your independent assortment, and that's basically all you need to know about meiosis. Go back and watch the mitosis video. It's going to be essential for you to know the difference. There's a table at the end of that that does give you the difference quite well. So basically I've been done from C.A.R. Entertainment. Hope you've enjoyed this video. I'll see you next time.